Today we'll be starting Unit 4, which is all about genetics and heredity. If you haven't already answered the Do Now question, why do cells divide? Pause the video and answer the question. There are 3 reasons why cells need to divide. The first reason is to replace old or damaged cells. When you get a cut, you clean the area and put a band-aid on. After a few days, your cut has healed. This is because new cells were produced. Another reason why cells need to divide is for growth and development. You should have noticed that you're a lot taller than you were from a few years ago, and that's because the production of new cells has helped in this process. The last reason why cells need to divide is because they can only grow to a certain size. If they get too large, it'll be harder for them to maintain itself by getting the proper materials it needs or letting out waste. Eukaryotic cells, which are seen in plants and animals, have a nucleus that holds their DNA, while prokaryotic cells, which are found in bacteria, don't have a nucleus, so their DNA is in the cytoplasm, which is the fluid part of the cell. Eukaryotic cells are bigger and more complex since they have many organelles or different structures inside that have specific jobs, while prokaryotic cells are smaller and simpler. Because of this reason, eukaryotic cells take longer to reproduce than prokaryotic cells. Let's take a look at the prokaryotic cell cycle. As you can see, only one parent is needed to reproduce. This type of reproduction is called asexual reproduction. From the picture, you can see that the DNA duplicates, or in other words, is copied, and in the end, 2 identical cells are formed. Let's observe how easily and quickly this bacteria reproduces within a day.
Bacteria reproduce very simply and rapidly by doubling their contents and splitting in 2. Just one bacterium, dividing every 20 minutes, could produce nearly 5,000 billion bacteria in one day.
In just one day, that bacteria reproduce billions of new cells. Since eukaryotic cells are more complex, our cells usually take about 24 hours to make new cells. Label this diagram and take notes about it on your paper while I go over the eukaryotic cell cycle. As you can see, the cell spends most of its time in interphase. Label A in interphase as G1. G stands for gap. During this part of interphase, the cell is growing and making sure that the DNA isn't damaged. Once the cell has done this, then signals from the body will tell the cell to move to the next part of interphase. Label B in interphase as the S phase. S stands for synthesis, which means to make or create, and during this phase, DNA is copied. The cell then gets signals again from the body to move on to the next part of interphase, which is G2. Label C in interphase as G2. During this phase, the cell makes sure that the DNA is copied correctly. It also prepares itself to divide. A cell spends most of its time in interphase in order to get itself ready before it divides. If something goes wrong and the cell divides, then it'll continue to create more damaged cells. What happens if there is something wrong during interphase? The first thing that a cell does is to repair itself if it finds any damages. If it can't fix the problem, then the cell goes through a process called apoptosis where the cell dies. This prevents the damaged cell from being copied even further. Sometimes the cell doesn't listen to the signals the body sends, and the damaged cell doesn't die but grows uncontrollably. This results in tumors and cancers. Now, a tumor is a mass of damaged cells that continue growing. If it is benign, the mass of cells continues to grow within a certain area of the body. But if it is malignant, then that is cancer. And that's when the cell starts to spread to different parts of the body. There are certain factors that can lead to cancer. If a person has a family history of cancer, then they have a higher chance of getting cancer than a person who doesn't have it in their history. Another thing that can cause damage to the DNA in cells is by exposing yourself to unhealthy things. For instance, if you go outside and don't protect yourself by putting sunblock on, the UV rays can harm your DNA and damage them. Let's take a look back at the diagram of the cell cycle. After the cell has spent most of its time in interphase, it will now start mitosis, where the cell divides. Label D1 of your diagram as prophase. This is the first step of mitosis. The second step of mitosis is metaphase. The third step is anaphase. The last step of mitosis is telophase. At the end, we have cytokinesis, where we will produce 2 new cells. Before we move on to discussing mitosis and cytokinesis, we need to talk about the important structures involved in cell division. Draw these structures and write down their functions on the right side of your paper. Before the cell divides, the DNA is like this messy ball of string. When the cell divides, the DNA needs to be more structured and organized in order to move around. So the DNA coils up into a strand called chromatid. These 2 chromatids are called sister chromatids since they are exact copies of each other. The whole X structure is called a chromosome, and the strands are attached at the center with a centromere. Another structure that's important is the centriole, which organizes and moves the spindle fibers. The spindles will pull apart the 2 chromatids. Turn to the back of your paper. We will write down what happens in each stage of the cell cycle and draw the cell. Use a different color for each box in the key to represent 2 different chromosomes, the centriole, spindle, and the nuclear envelope, which is a membrane that surrounds the nucleus. I'll be representing chromosome 1 with the color blue, chromosome 2 pink, centromere black, spindle red, and centriole green. As you can see from the graphic organizer, DNA replicates during interphase. Humans have 46 chromosomes or 23 pairs of chromosomes. Since we won't be able to fit all of the chromosomes in your drawing, we will only focus on drawing 2 of them. Like I said before, the DNA is jumbled up like a ball of string before the cell divides, so draw your chromosomes in an unorganized way. It also copies structures needed for cell division, like the centrioles, so draw 2 centrioles. At this point, there are no spindle fibers, and the nuclear membrane still surrounds the nucleus. After interphase, the cell will start mitosis. This is where the cell starts to divide. The first step of mitosis is prophase. Chromosomes condense, or in other words thicken, and become visible. They attach at the centromere. Centrioles will start to move to opposite sides of the cell. Spindles now form, and the nuclear envelope or membrane breaks down. The second step of mitosis is metaphase. You should be able to see that the chromosomes line up at the middle of the cell. So when you think of metaphase, think M for metaphase, M for middle. Centrioles are now at opposite sides of the cell. Spindle fibers connect to the centromere of each chromosome, and now the nuclear membrane is completely gone. The third step of mitosis is anaphase. You should see from the pictures that the chromatids are now being pulled apart. So when you think of anaphase, think A for anaphase, A for apart. The centrioles are at opposite sides of the cell. The spindle fibers have shortened since they're pulling the chromatids apart. The nuclear membrane is still gone. The last phase of mitosis is telophase. Take a look at the shape of the cell. This animal cell now is starting to elongate and look like a figure 8. The chromosomes are on opposite sides of the cell. The centrioles are still on opposite sides of the cell, so no change has occurred. The spindles are now starting to break down, and the nuclear envelope starts to reform again. When you think of telophase, think T for telophase, T for torn, since it looks like the cell is tearing apart. Now that the cell is done with mitosis, now it will go into cytokinesis. In cytokinesis, you will now have 2 new cells. Each cell will have its own set of chromosomes. It doesn't need to be organized anymore because we're done with cell division, so make sure your DNA looks like that jumbled piece of string. Each cell will also have its own centriole. At this point, the spindle fibers have totally disappeared, and the nuclear envelope or membrane is now fully formed around the DNA. Now you've been drawing the animal cell throughout this whole entire time. You could tell that it's the animal cell because it's circular. The 2 cells that are shown are plant cells. You can tell that they are plant cells because they're more boxy. For the most part, mitosis in plant cells and animal cells are pretty much the same, except for when it comes to cytokinesis. During cytokinesis, the plant cell membrane is too fragile to divide, so a cell plate has to form between the 2 nuclei. Eventually, this cell plate will turn into the cell membrane, and a cell wall will go around it.